Namaskar, this is Shadika Patana from the Department of Microbiology. The topic of the day is about the RNA structure and its types. So, in this video, we are going to learn about what is RNA, Swedberg coefficient, types of RNA, mRNA, its structural characteristics, its functions, hn-RNA, tRNA, its structural characteristics, its functions, the special CLOVE leaf structure of secondary tRNA, types, functions, structures of rRNA. So in the first place, what is a RNA? RNA is a single stranded molecule which is linked together by the phosphodiester linkage. The single stranded molecule is from the 3' to the 5'. The ribose sugar in the RNA has a phosphate group and a base within it. The ribose sugar has 4 nitrogen bases: adenine, guanine, cytosine, and uracil. In the case of RNA, you have uracil instead of thymine. RNA is synthesized from a DNA-based molecule called as RNA polymerase enzyme. It's because it is a DNA-dependent molecule, so it is called as DNA-dependent RNA polymerase. The process of this synthesis is called as transcription. Genetic code in RNA gets translated into amino acid, which gives rise to the functional product of the gene, which is called as proteins. In the case of eukaryotes, the RNA is synthesized inside the nucleus and gets transported to the cytoplasm for the protein synthesis. This protein synthesis can also be called as translation process. So now let's learn about what is Swedberg coefficient. Swedberg coefficient is invented by Theodor Swedberg, who's been awarded Nobel Prize for his work on colloid chemistry in the year of 1926. He is well known for his Swedberg unit. So what is Swedberg coefficient? It is a measure of rate of sedimentation of the molecule or a particle. It is the ratio of particle sedimentation speed to the centrifugal field which causes the sedimentation. The value of S of the sedimentation coefficient depends on some factors like mass, density, shape of the particle. The Swedberg unit is mainly used for the measurement of the distinguished ribosomes. The complex in the ribosomes, which include small and large subunits, have also rRNA and protein components in it. In the case of prokaryotes, the subunits are renamed as 30S and 50S, where here the letter S tells about the size of the molecule in Swedberg units. Let's now go to the types of RNA. There are 3 major types of RNA which is involved in the process of protein synthesis, which is mRNA, rRNA, and tRNA. The biological process like gene transcription, translation, and regulation takes place depending on the type of RNA which is being employed. There are 3 other types of RNA which is called as non-regulatory. It includes microRNA, smRNA, which is small nuclear RNA, and small interfering RNA. Now let's go into deep about what is mRNA, which is also called as messenger RNA. Jacob and Monod gave the name messenger RNA. It is said to be a long polymeric molecule which is made up of nucleotides. The mRNA comprises of 5% of the RNA cell. It is heterogeneous in size and the base sequence is attached with the phosphodiester bonds. mRNA provides a template for protein synthesis. The sequence nucleotides in the mRNA is complementary to the sequence nucleotides in the template RNA. Now in this point, I would like to remind you about the Evans-Charkov rule of complementary pairing. So this mRNA is said to have the complementary sequence of nucleotides in the template RNA. So if you see guanine in the template DNA, this mRNA will be having cytosine. This kind of pairing is called as complementary sequence pairing. The sequence carried on the mRNA is read in the form of codons. Each codon is made up of 3 nucleotides and the mRNA which is formed only after the processing of hn-RNA. Let us look in deep about the structural characteristics of mRNA. In the mRNA, you will find a 5' cap which is capped by the 7-methyl guanosine triphosphate. It involves the recognition of mRNA It stabilizes the mRNA by protecting it from the 5' exonuclease. You have a 3' polymer adenylate residues. You have a poly A tail, which prevents the exonucleases attack. You have untranslated non-coding sequence, which is present at the 5' end and the 3' end. The functions of the mRNA, the major function of mRNA include it providing a template for the protein synthesis. Heterogeneous nuclear RNA. In the mammalian nuclei, hn-RNA is an immediate product of gene transcription. It is heterogeneous in size. It is degraded by the nucleus, and only 25% of this hn-RNA is processed to matured RNA. The matured RNA has the following regions, which include a 5' cap, a 5' UTR, which is untranslated regions, a coding region, a 3' UTR, and a poly A tail. This is the structure of the matured RNA, where you have the 5' cap and a poly A tail at the 3' end. You have the coding sequence where the start codon and the stop codon is present. Now let's look deep into tRNA, which is transfer RNA. tRNA is the smallest molecule when you compare to the other RNA types. It has only 70 to 90 nucleotides. It is synthesized by the nuclear processing by the precursor molecule. They transfer amino acid from cytoplasm to the protein synthesizing machinery. It is easily soluble and so called as soluble RNA or sRNA. They are also called as adapter molecules because they act as adapters for the translation of sequence of nucleotides of mRNA in the specific amino acids. The important parts of the tRNA are its anticodon and the 3' terminal hydroxyl group, which can form ester linkage with an amino acids. The ribonucleic acids, and they are capable of forming the hydrogen bonds with mRNA. From the ester linkages with the amino acids, they can physically bring mRNA and amino acids together during the process of translation. They act as temporary carriers of amino acids, where they bring the specific amino acid to the ribosome based on the mRNA nucleotide sequence. Now let's look deep into the structural characteristics of tRNA. You have two structural characteristics. One is the primary structure and the other is the secondary structure. So the primary structure is a nucleotide sequence of all the tRNA molecules, which allows extensive intra-strand complementary, which generates the secondary structure of tRNA. The secondary structure is a single tRNA structure, which shows extensive intercellular base pairing and acquires a clove leaf-like structure. So this structure is stabilized by the hydrogen bonding between the bases and its consistent feature. This is the clove leaf structure of the secondary tRNA. You have 5' main arms, which is also called as loops, acceptor arm, anticodon arm, DNU arm, and TNC arm. Extra arm, it is also called as variable arm. When you look for the acceptor arm, it is at the 5' terminal end. It has 7 base pairs. The 3' OH terminal of adenine binds with the carboxyl group of the amino acids. The tRNA is bonded with an amino acid, which is the amino-acyl tRNA. The end sequence of the acceptor arm is unpaired with the cytosine. So the cytosine adenine bond at the 3' end, which is the CCA attachment, is done post-transcriptionally. When you see about the anticodon arms, it lies right opposite to the acceptor arm. It has 5 base pairs. It recognizes the triplet codon in the mRNA. The base sequence of the anticodon arm is complementary to the base sequence of the mRNA codon. Because of this complement sequencing, it binds specifically with the mRNA by the hydrogen bonding. DHU arm. So it has 3 to 4 base pairs. It recognizes the site for the enzyme amino-acyl tRNA synthetase, which adds the amino acids to the acceptor arm. Next comes the TNC arm. It is opposite to the DHU arm. It contains stereouridine, which binds the tRNA to the ribosomes. The last and the final extra arm or the variable arm, it has 75% of tRNA. It also has an extra short arm. It has 3 to 5 base pairs. Now let's look into the functions of tRNA. It includes the tRNA are the temporary carriers of amino acids. It transfers the RNA and it serves as a link between mRNA and the developing amino acid chain, which makes the protein. It is the key component of translation machinery where they read codons of mRNA and deliver it to the appropriate amino acids for the protein synthesis. Now let's look into rRNA. rRNA are the molecular machines that catalyzes protein synthesis. It constitutes of over 60% of ribosome by weight and are crucial for its functions. Their functions are from building to mRNA and recruiting. tRNA to catalyze the formation of peptide bond between two amino acids. The structure of the ribosome is determined by the three-dimensional shape of its RNA core. The proteins present in the ribosome stabilizes the structure to the interaction with the core. So now we look into the types of RNA, which is the rRNA. The prokaryotic ribosome and the eukaryotic ribosome. There are the two types of RNA. They both, like the protein ribosome and the eukaryotic ribosomes, are made up of two subunits called as larger subunit and the smaller subunit. Both the subunits join together on the mRNA translation. So the smaller subunit in the prokaryotes is made up of a RNA molecule about 1500 nucleotides in length and 16 years Wedberg coefficient with the ribosomal proteins. The smaller subunit has the sedimentation rate of 30 years, and this is paired with the larger subunit with two RNA molecules where you have 3000 nucleotides and 23 years, which is the Wedberg coefficient. The eukaryotic ribosomes is made up of 60 S and a 40 S subunit. There are two short RNA molecules, which is less than 200 nucleotides in length. The Wedberg coefficient of the ribosomes in eukaryotic cells is 80 S. The eukaryotic cells have the RNA in mitochondria and chloroplast in addition. The functions of rRNA. The primary function of rRNA is the protein synthesis. It binds the messenger RNA and the transfer RNA, which ensures the codon sequence of mRNA is translated accurately to the amino acid sequence in the proteins. The structure of rRNA include, it is basically a distinctive three-dimensional structure, which include an internal loop, A site, P site, and the E site. The internal loop creates a specific site within the ribosome, A site, which anchors the incoming tRNA charged with the amino acid. The P site binds with the developing peptide, where the E site, after all the peptide bond formation, it reaches the E site. This is the structure of the RNA, where you have amino acids, a larger subunit, a tRNA, and a smaller subunit. The A site, P site, and E site is present here. Thank you for your patience on my lecture. For further queries and doubts, comment below or mail us at sharp@gmail.com. Your mails will be responded within 24 hours of time. Thank you for watching. A like and a subscribe would be more encourageable. Thank you.